I'm Elijah. I'm at Hopkins, just right next door to Maggie, actually. And today I'm going to talk about probabilistic modeling of biological systems. So my talk's going to be maybe a little bit different than other people's in that I'm not going to talk about my stuff so much. So I'm going to talk very basic about why we model biology probabilistically, why we need to, what we hope to learn by doing that. And then I'm going to go through how we actually do the modeling. So it'll be like an introduction to my talk on Tuesday and other people who are doing biological modeling. So we won't have to do the methods then. You'll know everything there is to know. And in principle, you could run these simulations yourselves. Which brings me to kind of the first thing I'm going to talk about is I'm going to show a few slides just to do some introduction. But I'm going to go through some Jupyter notebooks that we have for doing this kind of analysis. So how many people in here are biologists? How many people are physicists? All right, at least you're intermixed, like you didn't segregate to one side or the other side. So this is supposed to be kind of informal. Really, I pulled a lot of material from my courses. So if you have questions, just ask so that everybody kind of can be on the same page about how this works. But what I want to show you first is this thing called Jupyter Notebook. So what do you guys use for data analysis? Biologists, what do you use? Python? Do you use Jupyter Notebook for Python? Does anybody use Jupyter Notebook? Does anybody not use it? So after this lecture, you should definitely use it because I think in the last few years, it's emerged as kind of the way to do biology and all kinds of numerical scientific analysis. It just has so many powerful features and a lot of people adding to it that it really helps. So I'm going to go through my tutorials in this Jupyter Notebook. And if we had tables, you could work along with me on your laptops. But I put up here, you can go to my website, I put up here a link to download kind of all the Jupyter Notebooks that I'm going to go through today. So if you want to go through them later or even right now, you can download them and follow along. And also on our website, we have kind of all the software that we use to do these simulations. So if you go to the software, our stochastic simulation software is this one called LMES. You can download it here. There's a VM that's set up, so you can just download the VM, run the VM, or you can download the code, compile it however you like. But that's what we're going to be using to do the kind of simulations we're going to talk about. So I wanted to show that before I kind of got started. Okay, so I'm going to talk about probabilistic systems. Can everybody hear me? Like I kind of walk around a lot, and the light is really bright. So like when I'm right here, I literally cannot see anything. It's very, very bright. All right, so this is kind of, I can't read too much of the text, but this is kind of how I introduce undergraduates or graduates. We have this kind of thought experiment. So this is kind of what we would like to do. We would like to have, cells, some kind of cells, who cares what kind of cells, growing in a petri dish, and you've programmed them somehow with some kind of sophisticated physics, followed by biochemistry, followed by cell biology, and you grow them, and you add a drop, and you can get them to do something, right? So we don't know how to do this. Nobody knows how to do this. You can make them photosensitive and flash something on there, and then they could reproduce that pattern. But we don't know how to have cells grow a particular pattern. And if you think about it, this is like insignificant compared to how real cells work. So real cells execute kind of much more complicated genetic programs than even this simple thing that we don't know how to do. So we need to understand how to do this, and we really need to understand the physics behind this. And it's different than physics in the rest of the world. Biology has its own physics. And we'd really like to understand kind of the rules behind this biological physics. So anyways, if you get bored, think about how to do something like this. Like we can do a circle. If you want to draw a circle, that's not too bad. We can do a ring, but we can't do a square. So if you want to have cells that can kind of grow into a square, now you have to break symmetry, and you have to have much more complicated logic in the interactions between the cells. So a circle is easy, but square, not so easy. And one of the things we need to understand in order to be able to do something like this is how cells vary, how the noise propagates through cellular systems, and how this gives rise to kind of what we call heterogeneity in cellular populations. And here's some just very classical examples noise in biological systems. You have E. coli. So even if you have the same gene, the same promoter, 2 copies of it in E. coli, some cells glow green, some glow red. There's all kinds of mixtures of colors because of just the normal stochastic variation in the population. Bacillus has a very famous program for DNA competence, which is driven by one of these noise-induced circuits. In mouse development, there's heterogeneous expression of nanog. And this is from Arjun Raj's very nice paper where they talked about whether cells can develop, say, a digestive tract or not, depending on kind of stochastic fluctuations during development. And so one question that we're interested in is how kind of this variation, how the microscopic noise in the system can give rise to kind of macroscopic phenotypic variability in cells. And one way we think about it is kind of this idea of a landscape, right? Biologists all know, like, Waddington's landscape. It's a very classical paradigm for developmental biology. But we'd like to really be able to map out, given a regulatory network, what are these stable states? How do cells transition back and forth between them? And that we don't know how to do. But we know how to approach the problem from stochastic physics. And this is kind of the way that, I don't know why these are all blue. The way that people think about this is that you start with some kind of complex network. So many different interacting species with lots of different kinds of interactions. And the way to think about this is it's in some kind of really high dimensional state space. And this space has a lot of stable, attractive points, and the cells move around in this state space driven by the dynamics of the regulatory networks. As people, we think about this in a much reduced landscape with hills and valleys and things. But in reality, it doesn't look like Waddington's landscape. It looks more like this, where it's very rough, with a lot of microscopic metastable states, and the cells are constantly moving around and jumping from state to state, even over kind of big barriers into what we would call different cellular phenotypes. Oh, and so this is a slide from Sui Huang, and one of the ideas is that this is a Waddington landscape that's been evolved for human cells, but sometimes cells get off track, and you can think about these cells as cancer cells or cells in some kind of non-native developmental pathway. And instead of maybe killing them, if you can force them back into the pathway, that's a different way to approach these kinds of problems. All right, so this is kind of, I just threw this in here because this is a slide I show sometimes, but so this is kind of a really complicated regulatory network, and this is just a cartoon. A really complicated regulatory network giving rise to some kind of a high-dimensional epigenetic landscape with several different attractive states. And we're going to label one of them a disease state. So if your cells are in here, you don't like it, you have a disease. So normally you take a drug, and the drug kind of kicks the cells out of this state, but they go somewhere else. And so you have some side effect to the drug because you've targeted the network just in one place very strongly. But what you would rather do is just remove this state and leave the other states the way they are. And maybe you could do that by making many small weak changes to the complex network. So if you knew how this network gave rise to this landscape, you could do that. But we don't know that, and we don't know the physics behind how this works. All right, so that's kind of why we do probabilistic modeling, so we can understand this mapping. And since, I don't know why it's flashing, but since a lot of people here are talking about phase condensation, we've been doing a little bit too. And so this is a movie, let me start it over real quick, showing the condensation of some kind of generic species, we call it X, from X1, which is kind of a non-condensed phase, into a condensed phase. And you can see that, that's really annoying, you can see that you get formation of these condensates and they move and diffuse. And if you watch them long enough, they kind of merge and they split and they evaporate. This is something we could do with kind of stochastic reaction diffusion. So this is what we're going to try to get to at the end of the day, is running a simulation like this. So we're going to talk about all the steps and the details that go along with that. All right, is there any questions about the introduction? Who's convinced that noise is important in biology? Who's unconvinced that noise is important in biology? Nobody? Okay. Well, some people probably are. But let's see how we go about modeling noise in biology. All right, so I've got these two Jupyter notebooks. We're going to go through. One of them is what I call kind of the theory, where we're going to talk about how stochastic modeling works. And then the second one is like the practical one. We're going to actually do some stochastic simulations and see how to run them and how to analyze the data. And if you don't learn anything else, at least you'll learn that it's very nice to use this Jupyter notebook. It's a very convenient way to analyze your data. All right, so let's get started. And has anybody used Mathematica before? So Mathematica, shift enter to execute the cell, right? The same way in Jupyter Notebook. So you're going to see me over here doing shift enter a bunch. And that's executing the cell in the notebook. So I'm going to run these cells. And the first thing we're going to talk about is kinetics. So a lot of people probably have biochemistry, right? Even the biologists have had biochemistry, probably. Has anybody not had biochemistry? All right, so in biochemistry you learn kinetics. And kinetics are just about the law of mass action. So how molecules react to each other depends on how much of the stuff you have. And this is, good enough for a test tube because you have like Avogadro's number of molecules in here. But what you probably didn't know is that all along when you do mass action kinetics, you're really doing the mean. So you're not calculating kind of the concentration change, you're calculating the change in the mean of the concentration over time. And for cells, for small numbers of molecules, that's not going to be good enough. So we need a different way to talk about kinetics other than just mass action kinetics. But before we do that, I forgot, we're going to do some little example. So anyways, we have a decay reaction, first order decay, and I'm going to write down the ODE that corresponds to that. It's just dx dt equals minus kx. And then, this is Jupyter Notebook. I'm going to do some numerical simulation of this, or numerical integration. So there's nice Python packages for doing integration. Scipy.integrate ODE. I'm going to give it a function that defines the derivative. It's going to calculate that. I'm going to plot it. And then we know the analytical solution. We can see the numerical and the analytical solution agree. That makes us happy. All right, any other questions on mass action kinetics? That's my introduction to mass action kinetics. No? Okay. All right, so kind of one step beyond mass action in terms of probabilistic is what we call the master equation. So I'm not going to go through much of the derivation or anything for the master equation, but you need to know that instead of kind of calculating the rate of change of things in concentration space, we're going to calculate the rate of change in probability space. So now we're looking at the probability for a system to have a particular number of molecules. And so what we would like to be able to do is kind of write down an expression for the probability to have 10 molecules, but we don't really know how to just write that down. Just like we don't know for kinetics how to write down a concentration of a function of time, we write down the gain-loss parts of the equation. So the gain of probability or the loss of probability. And from this set of differential equations, we can recover the original probability equation. All right, so I'll go through an example. So I'll use the board too. So let's just think about, it's a very simple first order decay process. So first order decay, is the same thing as nuclear decay that probably everybody learned about in high school or something like that. So say I have 10 molecules of some molecule that kind of decays away. And I want to look at, as a function of time, how many molecules are left. So it's a very simple question, and if you solve it with kinetics, what do you get? Who knows?
I think you use a black
Oh,
marker, maybe.
sure. The shape of the curve, if you're watching the decay, radioactive decay or something. Somebody just shouted out, everybody knows it.
Exponential.
Exponential. So normally we would expect to get some kind of exponential decay with some kind of e to the minus kt, where k is going to be our rate constant. But what happens if it's only 10 molecules? So if we just use this kind of law, then at some point I have 9.5 molecules, and that doesn't make any sense, right? That it's not physically possible that that's the right solution. So what should it look like? Intuitively, what do you think it should look like? And shout really loud. Somebody. For the probability distribution, you mean?
Yeah.
Yeah, but so what about in the real world? What would happen in the real world if you had 10 molecules?
It goes discretely down there towards 0
Okay, good. So it would go to 10, and then it would jump down to 9, then to 8, then to 7, then to 6. But would it do it kind of regularly? Yes or no? Who thinks yes? Who thinks that all of these intervals would kind of be the same? Like, if this is the half-life of 1 second, then each interval should be, 1 second or 1/10 of a second or something like that. Okay, why do you think that? Anybody? Who wants to answer? Nobody wants to answer. Who doesn't think that should be the case? Okay, so what should it be then? So these durations, what should they be? Anybody? I have a colleague, Richard Cohen, who told me one time that, when he teaches undergrads, and he said, when you get up there and you ask a question, you just wait. Like he said, you wait 10 minutes, 15 minutes, it doesn't matter. You wait till somebody gives you the answer. But I don't really have the capacity to do that. So you have to shout really loud. So who thinks, what do you think? What should this time be?
Exponentially distributed.
Exponentially distributed. See, somebody knows. Because these times are just going to be, they're a function of a random process, and the random process has no memory. Like an atom of uranium-235 doesn't know how long it's been an atom of uranium-235 for. All it knows is it has some instantaneous probability to decay, and if there's no memory in the system, there's no choice except for it has to be an exponential distribution of waiting times. So if we call this like tau versus the probability of tau, then this is going to be exponential, e to the minus k tau. So you might have to wait 0.1 seconds to get from 10 to 9. You might have to wait 10 seconds to get from 10 to 9. It's a random variable, a random process that depends on this rate constant. And so this is what we need to get to in biology is a way to talk about reactions. It's not happening at some particular number per second, but we need to talk about how long it's going to be until this reaction occurs in biological systems with small numbers of molecules. So this is what we do with the master equation. I'm going to show you some examples. The master equation is really, like I said, not one equation. It's really, for the case of 10 molecules, it's going to be, say, 11 equations. For an infinite number, or for a large number of molecules, it's going to be essentially an infinite number of equations. So we can't normally solve it like this, but for this case we can. So I'm going to integrate this master equation, and we're going to get back what we call a probability distribution. So now I'm showing x, the number of molecules, versus time, and the colors correspond to the probability of seeing that number of molecules at that particular time. So up here you can see it starts all off at 0 at 10 because we know we had 10, we put them there. But then it quickly kind of gets diffused because you don't know, you have a lot of uncertainty as to the state of the system. And then after a long enough time, you know that everything is decayed away, so it gets really high probability there again. So these are the kind of data that we get from a master equation approach. It shows us the probability to be in a given state as a function of time. And I have some other stuff here. I'm not going to go through everything because you can download it if you're curious, and I definitely don't have time. But we can calculate some statistics. So biologists know kind of statistics maybe as well or better than physicists. So we can calculate things like the mean and the variance of some of these things. You could calculate a student's t-test or whatever statistical thing you might be interested in. Let me see. And so again, from the simulation, we can calculate from the numerical integration of this master equation, we can calculate the mean. We can calculate the variance. And so we can again compare numerically and analytically our results. And for this very simple system, we can see that the numerically solving kind of this master equation is equivalent to solving it analytically. And then you can plot things more similar to how you might see that in biology, where you might have kind of a mean plus or minus some error bars or some variance or something like that. All right, so this is the master equation of kind of a very simple birth-death process, of a simple first-order decay. We do the same thing for a simple birth-death process, we call it, where we just have some molecule being created, some molecule being destroyed. What's the difference between this birth-death process and kind of the decay process? So they're kind of very different because one converges to zero and the other does not. So with a birth-death process, you're going to have a long time probability to be in a non-zero state. And so that's what we're going to be interested in. And if you hear like a physicist say this is going to be the stationary distribution, that means it's a not changing distribution with time. So if it's stationary, it means it's not moving. So in the long time limit, these distributions are going to be called stationary, where they're not changing in time. So we can do the same thing we did before, run some simulations, or rather numerical integration of the master equation. And you can see that we start in some out of equilibrium state, say at 0 molecules, and then we converge into some kind of distribution. But if you look at a vertical slice through here, you can see that it's not changing with respect to time. It just becomes some constant distribution. This is what we call a stationary distribution. And like we did before, we can look at the mean and the variance of these distributions. And so what we can see is that, so I forgot to tell you that, so even now we've got to a point where it's becoming very hard to get an analytical solution for the time-dependent distribution. So we can calculate what the stationary distribution should be. I gave away the answer. I wanted to have a question, but the answer is that the stationary distribution is what's called a Poisson distribution. And Poisson distribution is a special distribution. Who knows something about Poisson distribution? What's special about Poisson distribution?
Is it a memory loop?
Oh, that's exponential. Variance equal to mean. So everybody knows that because if you do, say you're doing single cell imaging and you calculate the mean of your cells, you calculate the variance of your cell. If they're the same, then you say, oh, there's a Poisson process driving the noise behind the system. Or if it's more than the variance is more than the mean, then you say it's a system that has more than Poissonian variance. So something else is driving it. So it's kind of a well-known distribution. But anyways, what we can see is that eventually the time-dependent distribution converges to whatever we would expect from the analytical solution for the stationary one. But we can easily calculate the time-dependent distribution numerically, whereas we might not be able to do that analytically in the general case. And so even what I was showing you here, and like I said, you can go through the notebook if you're interested later on, was what we call integration of the system of differential equations. But this gets very hard for systems of any kind of complexity because the size of the space just goes up so fast. So instead, we oftentimes use something that a lot of times is called the Gillespie algorithm. which is a way of sampling kind of random trajectories of a particular system and then combining them to get statistical information. So this is called Gillespie algorithm, and it's actually very simple. The only thing you have to do is, we go back to this same picture, you have to know that the time between reaction events is exponentially distributed. You can calculate what this rate constant for the exponential distribution is, and then you randomly sample a time to a next reaction. And so you just do this over and over again. You calculate the time to the next reaction, you update your state. Calculate the time, update the state. Calculate the time, update the state. Just for example, like our software that we have runs this. The inside loop is kind of very small, and it runs maybe 20 million reactions per second. So a lot of time is spent just generating these random exponential distributions and then updating some table of the counts of species. But you can generate very long, large trajectories, and a lot of them very quickly. All right, so we did one here. If you want to see, this is basically the Gillespie algorithm for this very simple system. And this is kind of a random trajectory we get out of it. If I run it again, you can see I get a different trajectory. If I run it many times, then I get a different set of trajectories, each which are kind of perfectly valid, but none of which really describes the full properties of the system. What you need is many of these trajectories, and you can see now that if you were to do this many, many more times, it would map out the same probability distribution that I showed before. And that's how we're going to calculate probability distributions for really complicated biological systems. All right, is there any questions? Yeah.
Maybe you can mention as a rule of thumb, like the ranges where you can, for 10 molecules, so you have motivation to use stochastic and master equation. For 1,000, a million, Avogadro number, probably like killing a fly with a cannon.
Sure. Yeah, so actually I was going to skip this, but I'll just show it that we can also calculate, given a particular, say, time interval. So we go back to our example. If you say that I want to know what's the probability in 10 seconds that how many reactions have occurred, you can calculate what that distribution should be, and that actually also will be a Poisson distribution. So you could calculate with any kind of interval of time how many reactions are going to occur and what's the mean variance of that distribution. So that's what I show here, is that if you have one molecule, then you can see the blue curve kind of has a pretty wide range of the number of reactions that might occur in a second. The more molecules you get, you can see that the mean is shifting up, but then it's also kind of getting more and more compressed. And once you get to, say, see if this runs, here it would be for 100 molecules. And once you get to 1,000 molecules, now you can see that this comparison with just a Gaussian distribution, which would be what you would expect from a set of ODEs. By the time you get to 10 to the minus 15th in your probability, it still is in pretty good agreement. And when you're up to, say, 100,000 molecules, it's basically indistinguishable. So, if you have 100,000 molecules, you probably don't need to do stochastic simulation. But if you have 100, you might need to. So somewhere in that range is where it starts to matter. All right, and so the simulation that I showed at the very beginning, everything that I talked about up till now is kind of for like a test tube, where it's well stirred, things are happening in there very quickly, molecules are having many, many collisions before they react, and they're losing memory of their previous location. But we also have a method called reaction diffusion master equation, which is the same kind of a thing, except it divides it up spatially on a grid, and each of the subvolumes is itself a well-stirred simulation. And then molecules can jump from subvolume to subvolume using some rules to describe kind of diffusive probabilities. And so when we try to do cells, which of course are not just a big bag of stuff, we tend to use something like this to look at the spatial localization. In many ways, a lot of the theory and the statistics are the same between these things. All right, does anybody remember what time I started?
Started 20 minutes earlier or something like that.
I can't remember.
Yeah, I think 20 minutes ago, 15 minutes ago.
What time was the next break supposed to be?
Just now.
Just now. All right, so let's
We are a little bit in front of the...
say again, sir.
The break is supposed to be in 5 minutes, but then we started a little later, so 15 more minutes
Before the break.
before the break, yeah.
Okay. All right, so does anyone have any questions about this before we kind of get to the simulation part? How about questions about Jupyter Notebook? How about questions about anything? No? Okay. So the next section we're going to talk about is going to be kind of the software that we have for doing these stochastic sorts of simulations. Like I said, we call this software LMES. It does both the chemical master equation and the reaction diffusion master equation. And you can go to the website and download it, like I said. And it's pretty straightforward to get it up and running. But what I want to show you then is using it to calculate some systems that might be relevant for kind of people studying sorts of genomic dynamics and architecture things. All right, so I'm just going to get this simulation started. And then the first thing we're going to do is simulate what people call the genetic toggle switch. So the genetic toggle switch is a very classical example of gene regulation in biological systems. And it basically is a gene that has two genes that are share the same promoter. And so in this particular model of the toggle switch, only one of the genes can be expressed at a given time, and that's determined by whichever of these transcription factors is bound to the promoter. So when gene A has been expressed, it's switched to kind of the high state. There's a lot of gene A around. It will bind to the promoter, and then that will continue expression of gene A and inhibit the expression of gene B. And the reverse is true, that when gene B is being highly expressed, it will bind to the promoter and inhibit expression of gene A and only promote the expression of gene B. And so the reason this is called a toggle switch is that what will happen is it will randomly transition between these two states, the state of high A expression and high B expression. And this is a nice model for kind of these epigenetic decisions that we talked about at the beginning, and that you have a state where it's high, say, gene A, and there's some kind of a barrier, and then another state where it's got a high gene B expression. And in general, the transition back and forth between these two states are called rare events in the kind of language of dynamical systems theory, because the transitions are rare relative to kind of the normal relaxation of the system. So you'll see that the genes are going to fluctuate a lot within a barrier, but then every once in a while it'll make a transition into the other basin. And that's what we call a rare event. All right. So the first thing we need to do is to write down the biochemical model that describes the system. And so we're going to use this language called BNGL, which is kind of a nice way for people to write down models. There's various ways in the software to actually go about expressing a genetic or kind of a reaction model, but BNGL is probably the easiest for a person to look at. It was created originally by the BioNetGen people for doing kind of rule-based modeling, but we use it for this too. And you can see it begins by just defining some parameters. So you have a model, you have parameters, of course, that define the rate constants, and we're going to have some rate constants for dimerization. So A and B dimerize, so we need rate constants for that. Binding of the dimers to the genome. So if you remember from the little schematic, then they can dimerize back and forth, and the dimers are what bind to the to the operator site. And then we need rate constants for kind of the birth and death of the proteins. So in this case, we're going to have, say, the birth of the protein is just some parameter that we're going to pass into the call, something we call theta. And the death rate is going to be kind of slow relative to that. So then we define kind of our molecules. So we're going to define operator. We're going to define a bound operator. And we're going to define A, B, and A dimer, B dimer. And then we're going to have only one operator. We're going to start it in the bound state. So we're going to start operator bound to it with an AB dimer. We're going to start with 4 molecules of A and 16 molecules of a dimer. And then we just specify the reactions. So just like we said, we're going to have A dimerizing, B dimerizing, A dimer, B dimer binding to the operator, and then the birth, the promoted birth, and the 2 degradation terms. All right, so then we need to import kind of this reaction model. And then so we do that using kind of this little tool, Gen import, give it the BNGL file. We're going to pass in some particular theta that we're interested in. And then we're going to set some properties. So we want to run a simulation, and we're going to run it for 100,000 seconds. And then we're going to write the state of the system out, say, every second. So we're going to set that up in the input file. So, it's really hard to organize everything in just one big text file. So we use these files called HDF5, which let you store binary data. So it stores essentially a lot of matrices and things in here. If you want to see any more of this, the user guide for LMES, which is here in the tutorial section, has all the details about what these matrices mean and how to specify them and things like that. Back. And then we can run the simulation by just essentially calling the LMES program, give it the file name, and we're going to run, say, 10 replicates. So like I showed in kind of the examples for this Monte Carlo sampling, we need to generate many, many trajectories, each of which is kind of an independent realization of what might happen in this system. So here we're going to run 10 trajectories. And you can see it only took a few seconds to run. We're going to then open the file. And again, these are binary files. And Python has a nice package called H5Py, which you can use to read these binary files. And anyways, even if you're not using this, like HDF5 is a really nice format for storing data in. Like a lot of times I'll get text files or Excel files from people, and it'd be like 100 megabytes, whereas if you had a nice binary file, it might be just like 1 megabyte to store that. So you might want to at least realize that's out there. We're going to load the data. We can see that we're going to have an array now with 100,000 entries, one for each second, and we're going to plot these things. So now what we can see from the plot, so now I'm going to plot, for example, O, and O doesn't look like much because it's just rapidly jumping back and forth between the free O and the bound O. But if we look at the OA2, which is the O state bound by the dimer, then you can see that it will jump up and then it'll go off. And this is indicating that the switch is, or the system is jumping between these states that we called them, the high A state and the high B state. And if you look at the OB2, you see kind of the anti-correlated behavior, where when it's in one state, it's not in the other state, and vice versa. And you can see the same thing in the concentrations of A and B, where at some point you have high A and low B, and at another point you have high B and low A. And this is kind of very indicative of this jumping, this transitions between metastable states that we talked about. All right, so now I'll introduce just a little bit of terminology for people who might hear this and might need to know what it means. It's not often very easy or compelling to kind of show lots of different species. So a lot of times we'll do what is called make a reaction coordinate or an order parameter or something that describes just the system in, say, one dimension in a way that's more informative for us. So here we're going to do the same thing. So we don't really care about the number of A's or B's. What we care about is the switching between the states. So we're going to define an order parameter that's going to be, say, the sum of all the B molecules in the system minus the sum of all the A molecules. So if we're in a high A and low B, this should be negative. If we're in a high B and a low A, it should be positive. And that'll allow us to more easily look at kind of the transitions. All right, so let's do that. So now I'm going to load all of the trajectories, all 10 data sets, and then I'm going to plot a probability distribution, a histogram, of the probability to have a particular value of an order parameter. And now you can see that we see these two states that we talked about before, and additionally we can see kind of this minimum between the states, which here is going to be, say, 10 to the minus 4. And so the value of this minimum indicates something about the first passage time, how long it takes to jump from one system to the other system. Because if you get right to the midpoint of this transition, then you essentially have a 50-50 chance of falling one way or the other way. So the time to reach kind of this minimum is a very key quality of these sorts of switching states. We typically call this like a mean first passage time or a mean switching time or something like that. And here it's not very strong. So good rule of thumb is if you have 10 to the minus 4, if you flip that, it's going to be 10 to the 4 seconds for first passage time, which is pretty short for this system. All right, is it time for a break?
Yes.
Okay. All right, so we'll pick up in 10 minutes. Till we get started again. So people came back, so that's something. We should come back with like beers, though. I think they have an open bar still, right?
Okay, so 5 more minutes.
Okay, so we've calculated kind of this probability distribution in the order parameter space, but we also might like to see it in kind of the full state space, we would call it. So in kind of the real two-dimensional space of the system. And so in order to do that, we're going to need to generate more data. So we generated, say, just 10 trajectories, but to do large searches in kind of really high-dimensional spaces, you're going to need a lot of data. So I'm going to just talk for a few minutes about how to do that and what it means. And while I'm talking, I'm just going to run another simulation. And so now I'm going to do 90 more trajectories. So I'm going to do replicates 11 to 100. And it should take about 2 minutes to run. And like I said, I'm just running all this stuff on my laptop, right? So like these simulations, the CME simulations, they're just very fast. It's just, you can do 20 million reactions every second. We commonly use parallel computers, and maybe we're running trillions, literally trillions of reactions to generate these things, because it can churn through reactions just very quickly. And it's going to run here in about 2 minutes, and you're going to see that it's going to generate a 350 megabyte file. So in like 2 minutes, I'm going to generate 350 megabyte files. And actually, this was a real problem for us for years, is that we would run these long simulations, and they would generate so much data that copying the data back from the supercomputers took as long as it did to run the simulation in the first place. And then once we got it back, it would take that long also to analyze the data, because you were limited always by the speed that you could read the data from the disk that you stored it on. So some years ago, we started to look at what people call data-intensive computing. And so this is a technique kind of pioneered by like Google and people who started to first index the internet. And they don't save it all on one disk, and they don't buy really expensive fast arrays. They just save it to like thousands or hundreds of thousands or millions of disks in different computers all over the place. And then the key innovation is you don't bring the data back to one computer to analyze it. You send the code that you do the analysis with to the data. So you take whatever code, your script that you're going to run, you send it to wherever the data is, and you run it in parallel, and then you just bring back the summary back to the system. And so we have this framework that we've used along with what people call the Spark. There's a Spark environment, which is kind of like a next version of MapReduce, which is Google's kind of original thing. So we have some framework that we call BioSpark that allows you to analyze kind of numerical biological simulation data in this data-intensive way. And now, you know, data analysis is very fast compared to what it is. And you can get, you know, if you have a little cluster of these things, you can get 80 terabytes for not that much money, and it comes with kind of processing power built in. So you can analyze simulation trajectories pretty quickly if you're willing to put in time to look at this new thing. Anyways, we have this BioSpark software, and it's also out there for download on our site, and we have tutorials that kind of show you how to use it and get started with it. So if you're really ever limited by time to analyze data, it doesn't have to be simulation data. We've also used it to analyze, a while ago we did some analysis of microscopy images using like neural networks and things, and we had a lot of data for those, and it was very useful to very quickly process these data using that approach. But anyways, you can see that this finished now. We have 90 more trajectories. It's a 350 meg file. So one danger actually with running these softwares, if I had accidentally set like a really low time step, I could have filled up my disk in like 3 minutes. So you can be a little dangerous, and we have had undergrad students fill up the cluster because they have a time step too low. So you have to be a little cautious. Anyways, I'm going to reload the data. So now I'm going to load data from 100 data sets with 100,000 steps each. So now I'm going to have 10 million data points to do my analysis with. And we can plot kind of the joint probability distribution now. So the probability to have A and B numbers of these molecules at the same time. So let me plot that. And this is kind of what it looks like for this model of the genetic toggle switch. You can see that there's one stable state here, metastable state, another metastable state here, and in between is this transition barrier, transition region where the system switches when it stochastically jumps between these two metastable states. All right, questions about this so far? Yeah.
So is P of AB your probability?
Yeah, it's a joint probability to be in A and that value of A and that value of B.
Okay, I guess I'm a little bit confused because it looks like you have negative numbers there.
Oh, yeah, this should be log. This is log 10 of probability.
Okay.
Yep. Okay. All right, so then I wanted to have some extra time, so I'm going to show you real quick some other work that we've been doing. So up until now, what I've shown you is just generating trajectories and calculating the probability distributions from these trajectories. And normally that's fine. It can be slow for systems like this where we have these rare events, because if you look at this probability distribution, you know, most of the time I'm just here or I'm just here, but what I care about is here. And so I spend very little time in the actual part of the phase space where I'm interested in. So it's not really an efficient use of computing time. Who cares about my laptop? But if I'm spending, you know, thousands of CPU hours on a complicated model, I don't really like to waste that much time. So we've been working on some of these enhanced sampling methods that people have developed for kind of improving the way that we run these sorts of simulations. And actually that's why we call it LMES, is because it was Lattice Microbes, which is the software that was developed in Zan's group, but we added this enhanced sampling features to it to allow it to run these enhanced sampling simulations kind of using this underlying framework we had for generating these trajectories. So let me just show you real quick some of the details of this enhanced sampling work. So I don't have this formatted, so it's going to be a little ad hoc, but let me see if I can find the figure I wanted to show. Yeah, so the basic idea is behind this and a lot of different enhanced sampling methods is that you've defined some kind of an order parameter, which we already talked about. So we have an order parameter that describes how the system moves in one-dimensional long phase space. And then we saw our probability distribution where we had kind of a minimum in the middle. So what we can do is we can divide this system up by these interfaces, or these phases that we call them. And then if you can calculate statistically what's the probability of going from this phase to this phase, or this phase to falling back into the basin, and you can do that for each one of these basins separately, then you can combine statistically these weights to obtain the kind of original probability distribution. And then you're not spending any time simulating these regions, or very minimal time. You're spending most of the time just starting trajectories, launching them from different points on this interface and seeing if they succeed or fail, and tracking all of this individually. So you've translated the problem from just running a bunch of simulations and seeing what happens to starting literally billions of trajectories at different points on this interface and tracking whether they succeed or fail. But it can turn out to be a much more efficient way to calculate data. Let me just go to the end here.
Is this just for calculating the relative probabilities, or do you also get the time information for how single particles explore?
You get the first passage time. So originally it was developed to obtain the first passage times, which describe kind of how long it takes to go from one of these metastable states to the other metastable state. But with those weights, you can also reconstruct the probability distribution by saving probability distributions at each interface and then reweighing them by the original weights they calculated. But it is only a stationary distribution, so it doesn't calculate kind of, you've got a time-dependent process. People have worked out some theory for that, but our model doesn't have time-dependent non-equilibrium properties. So as long as you have a stationary distribution, it's good. But if you have some kind of rate or something that changes in time, then it wouldn't be able to calculate that. Does that answer your question? Any other questions? Oh, before we go? Okay. So anyway, so this is called what people have called forward flux sampling, because you're calculating. Let me get back to my figure. You're calculating. Where can I not find it? You're calculating the flux essentially out of this basin through, and processing it forward across these interfaces. But like I said, you have to start lots of individual trajectories from each one of these interfaces to calculate it. And so it was a little unclear, like how many. So if you get to interface 0, do you do 100,000? Do you do 1 million? Do you do 10 million? How many do you do? So we worked out kind of the theory describing the error characteristics of the system. So essentially, how many, if you wanted to get a 1% error goal 95% of the time, then how many trajectories should you start at each one of these interfaces? And then it turns out that if you can do that, you can also optimize it for the cost. So we can minimize the total computational cost to get a particular error. So say you want a trajectory that's 1% error with a 95% confidence interval. It'll tell you exactly how many trajectories to start at each interface to minimize the total computational cost to go with that. And if you use a method like this compared to what we call direct sampling, you can get really incredible speed ups. So the speed up scales, of course, with the rareness of the event. So like for some systems, the toggle switch actually that we calculated, we're getting 300 or 400 times faster when you do the enhanced sampling versus kind of the direct sampling method. So there's really a lot to be saved when you don't have to worry about just calculating the boring stuff all the time. And so this also, we have tutorial on. It's in the software. You can download it and use it.
I have a
Yeah,
question.
sure.
So does this make the assumption that all points at a particular reaction coordinate are equivalent to each other?
It makes the assumption that they all behave as a Bernoulli process. So it samples all of the states separately, but it does make the assumption that they all can make it to the next interface with equal probability, with a probability that's Bernoulli distributed. So they don't have to be in the exact same state, and it will sample them according to their relative weights in that state. But to get the correct number of trajectories, it assumes that they're all following a Bernoulli process. And that may not be true. Like it may be that a certain place in the reaction coordinate is more likely to make it to the next one than the other, and they don't relax fast enough. And then that can throw off the calculation of the weights. That's right. Okay, any other questions? All right, anyways, our thing is called FF Pilot because we do a pilot run. We do a very short run across there to calculate preliminary weights, and then we use the weights to do this optimization problem. Okay, anyway, but when you just run a simple thing on your laptop and it takes 5 minutes, it doesn't matter if you run it direct sampling or forward flux sampling. Okay, all right, so this is, everything I've done up till now is the chemical master equation, so this well-stirred approximation. Now I'm going to be done with that. But before I'm done, are there any other questions about chemical master equation or well-stirred processes? Okay. All right, so I'm going to talk about RDME. And specifically, I'm going to talk about this phase transition model that we've been working on. So not very long. So, we haven't been working on phase condensation, but TJ's at our institution, he had some really cool data, and Crosstown had some really cool data. And so we wanted to see if reaction diffusion models, without kind of this detailed structural information, could provide any insight into these phase condensation problems. And if so, what could you learn from these problems? So I'm not going to talk about the results because I'll talk about that a little bit on Tuesday. But I'm going to show you kind of how to set up the models that we're running and how to run them and what kind of things you can get out of them. And so it's all kind of originated actually for work that we've been doing about kind of clustering of receptors, but we didn't call it kind of phase condensation. We just talked about it as clusters. And it turns out that any kind of a reaction diffusion model, if you have kind of some kind of positive recruitment of this new interacting version of the molecule, and that version has a lower diffusion coefficient, you can get localization and condensation of things. So I'm just going to show you a very, very simple model. So we just have two proteins, whatever, X, and they can form another species, X2. But X2 is not really a dimer. So it's just two kind of interacting things. I just put X2 here because that's what we use, but it doesn't necessarily mean that it's a dimer. And then if you have auto-recruitment such that this set of interacting things can bring more of these proteins into the interaction state, then you have enough essentially to get a phase transition. And so I'll show on Tuesday, you can show that in the dynamical system you have multiple fixed points, and these correspond to the different phases of the system. But what I'm going to show today is just how to run it. And we're going to do the same thing that we did before. So we're going to have a BNGL file which describes kind of this process. And you can see this one is very simple. So literally that's it. So you have 4 rate constants, two species. Oh no, so I'm sorry. So the one thing I didn't show you before, and BNGL is good at, is it's good at doing kind of complexes. So you don't have to define each thing kind of individually. You can just say there's one species, and it can form a complex, which is indicated like with this dot. So this is their syntax for saying that two things are kind of bound together. So we literally have one species. We have this dimerization that's not really a dimerization, and then we have this auto-recruitment. And that's it. So the same thing that I did before, I'm going to generate a simulation input file by importing this BNGL file. So these ones are slower. So the RDME is definitely slower than the master equation, the CME. So we're going to run it for 100 seconds, and we're going to write out the lattice and the state every second. And then we specify a couple other things because this is a diffusion model. We need to specify the boundary conditions. So we're going to set reflecting boundary conditions. You can do absorbing. You can do constant concentration boundaries. You can do constant gradient boundaries, any kind of different boundary that you might be interested in. And then we're going to have to specify the lattice size. So we're going to do a 30 by 30 lattice, 30 by 30 by 1, like a pseudo 2D model. And then we're going to specify the two diffusion coefficients. So the first diffusion is 5 times 10 to the minus 10th meter squared per second. Who knows what a protein diffusion constant is? Anybody? 1 micrometer squared? That's a little slow. Maybe.
Between 1 to 100.
Yeah, somewhere between 1 and 100. GFP, I think, is 80 in vitro. On the membrane, they're going to be much slower. So this is a little fast for a protein, but anyways, it's just a parameter. And then we're going to have a diffusion coefficient for the other species, which is kind of the species that's in some kind of weakly interacting state with a bunch of other of these things. And we're going to set it to just be relatively slow. 15 times slower, 50 times slower. All right, so we're going to run the simulation. And so my laptop has 4 cores, so I'm going to run 4 replicates because I get them all for free in parallel. And it should take like 20 seconds or something to do this simulation. All right. So now we're going to load the data. And so we need to load, now it's a little more complicated data, so we have to load the entire lattice at each one of these steps. So, like I said, if you want to ever see the details, we have all these getting started guides and things to show you how to do the calculations, how the data is stored in the file. But for now, I'm just going to load the data into a lattice. I can sum over the whole lattice, like we did, like for the chemical mass equation. And the interesting thing to see is that you can't see it here necessarily, but now we're going to go to some particular steady state for the uninteracting species and a steady state for this interacting species. And then I can view some of these time points in the lattice. And so this is what you see. So at kind of 0 time, it's all in this is, I think, only showing the interacting species. So at the 0 time, it's all in the uninteracting state. And then, very quickly, you form these clusters. And then, of course, since this is like a time-resolved lattice, it's most interesting to look at the movie. So we're going to make a movie of this. And this might take a few seconds. So it's got to draw all these frames. And this somehow actually it's slower to draw the frames of the movie than it is to calculate the simulation itself. So I don't know what to say about that. But does anybody have a guess as to what you might see? No guesses? All right, so finished. Nice program called FFmpeg that takes bunches of frames and puts them into a nice MP4 movie for you. I open it up, and then this is kind of what we get. So we can see that it's going to show you here kind of the overall concentration of the uninteracting species, the interacting species, and then the lattice for each one of these two. So at time 0, everything's just kind of uniformly distributed in the lattice. And then very quickly we see that we get a cluster of this condensed state. And this thing forms and diffuses. And of course this is just a random trajectory. But if you look kind of towards the end, it looked like it was starting to maybe split and then it merged back together. And you can see all kinds of very interesting dynamics in these processes. So last night I ran a longer one just so we could do that. So a longer one I think takes about 45 minutes on my laptop. Let me find it. And it's a bigger lattice. So now this is a 100 by 100 lattice, but it's essentially other than that it's exactly the same as we just ran. And you can see very early on at the beginning, before you have any kind of structure, you form these very small clusters. And then you can just watch the dynamics, and you can see that they're diffusing, they're merging. I think if you watch this one, it evaporates after some amount of time. Like this one, I think almost splits, and then it goes back together. So you can see a lot of really interesting dynamics just from this very simple reaction diffusion model. And again, we don't have any details of intrinsically disordered domains, amino acids. We don't have anything like that, so we can't answer questions related to those types of problems, of course. But we can run very fast, and we can run many different sorts of simulations. So what I'll talk about on Tuesday is kind of some of the work we've looked at in terms of how these properties, or these little condensates, if you want to call them that, behave in different parts of the phase space. So we can change the concentration in here, we can change the diffusive properties of the system, we can get to places where it is condensed and it's not condensed, we can get to places where it jumps back and forth between condensed and uncondensed, because this turns out to be, if you do a dynamical system analysis of this, you have multiple stable fixed points, just like we saw for the toggle switch, that the system will jump back and forth between these stable states. So I could talk more about that. I'll talk more about that on Tuesday. But anyways, just so you can see, we can run more and change. Let's change the diffusion coefficient of, let's say, the condensed species to be even more slow. Does anybody have a prediction? What would you expect? If you're slowing down now diffusion of the condensed species. Faster clustering? That sounds reasonable. Any other ideas? Larger cluster. So the interesting thing about our system is that, first of all, there's fixed numbers of molecules. So the interesting thing actually is that if you decrease the diffusion coefficient, you tend to get smaller clusters because the things can't escape the cluster. The flux out of the cluster is lower because the things are diffusing less, whereas the flux into the clusters is similar. So if you look at the movie, or if you look at the individual frames, you can even see, that that's happening, that these are much more condensed sorts of clusters than in the other case. But because they move slower, they split more often because they're not interchanging with each other quite so frequently. But like I said, so this is kind of the limit of what you could do with a laptop, 100 seconds. But in general, if you can run them on kind of a cluster, you can do a wide exploration of the parameter space of this system. And the software makes it pretty easy to just do lots of different parameters and things like that. All right. Any other questions? Yeah.
Can you treat the diffusion count, I guess, close to 0 and
Close to 0 for the interacting species? Sure. How about like 10 to the minus 15? So, I mean, it really makes nice, interesting movies to look at, and you can kind of look at the dynamics of this process. And we've been doing some other stuff, like I said, where we're looking at kind of the FRAP properties of these clusters, how they change with different regions of the phase space. But you can see it's very straightforward to run these sorts of things, right? It's just, I ran a few cells, generated a few files, ran for 15 minutes, and you start to see how these things look. Do you have a prediction for this? What do you think? No clustering. Why do you think that?
one component is fixed. That's down the reaction.
Ah. We'll see. Let's see. After this, I'll show you. I'll do one where see if I can get to the region where we jump back and forth between the across the phase transition. I don't know. So it's very strange. So you have formation of a cluster, but it seems to grow kind of in one particular direction.
Oh, that's bizarre. That's actually pretty interesting. I would say that.
It could also be it could be a mistake. So, of course, the software it's a lattice software, and each subvolume kind of has a limited number of molecules that it can store. So if you're condensing things and they keep going into that subvolume, the way that it takes care of that is it moves them to the next subvolume. So probably that's what's happening, is that it's filling up the lattice site, and then if a particle condenses, it's moving it to the next nearest lattice site, neighboring lattice site.
But always in the same direction. Why not like in?
Yeah, it probably starts the search, like at the top, and searches around for an open lattice site. So it probably always puts it in the same place. And then as soon as you have a new site with a condensed species, it immediately gets filled up and starts happening again to the next one. If you look at the log files, it will tell you that this is happening, but I just didn't look at them for this tutorial. You just dump them to erase them. So that's interesting. Let me,
You need to randomize the search for the open subvolume.
in general, it doesn't matter too much if they're diffusing, because they randomize themselves over short periods of time. So let me, this is our little phase diagram. Let me get to a region that might show more interesting dynamics. Oops. So let's try like 4 times 10 to the minus 11.
It stops doing this once you make it big enough, right? I'm assuming. Once the diffusion rates are much, much faster than the reaction rates.
Yeah, that's
There's
right.
no.
Once it has ability to move before being overloaded with reactions, that wouldn't happen.
Right.
Or you could increase the limit, but in this case, if it's 0, they're all, the end state for that simulation actually is everything in one lattice site, and that's not really interesting for us. So. Of course, now I overloaded my laptop. So here we go. So in this one, you can see it forms some of these clusterings, but the diffusion coefficient is much faster, so the clusters are much more spread out, but you still have this stable state. So it's still stable in this kind of condensed sort of state, but it's a much bigger sort of area that it encompasses. So let me see if this one finished.
So what's the parameter that was changed in that? Was it a K that was changed or was it a D?
It was a D. And I ran it for longer, I think, too. But I think I increased the diffusion coefficient. I didn't write down what I wrote it at, saved it as. But it could be this is the same one that I ran just now. But for longer. So this one looks the same sort of thing where you start to have these much more diffuse sorts of clusters, yeah. So if I were able to run it for longer, you could see there are some cases too where you can jump back and forth between the totally diffuse state and the condensed state, almost like you're having a sort of phase transition in the system. But anyways, these are just little toy model to look at. If you want to run these, like I said, you can download the notebooks out there. Okay, I think that's basically all I wanted to say. Like I said, everything's out here in tutorials. If you want to run these things, we spend a lot of time putting the tutorials together. You can do the stuff without too much hassle. You can download the virtual machine. If you don't have a good machine to run these on, you can run them on Windows or whatever else if that's what you use. Any other questions? Anything? Yeah?
It seems that there's a steady state
dimer ratio. Is that different how the diffusion coefficient or that's just the result of ODE?
Does it depend? What does it depend on? What?
The diffusing.
Oh, yeah. So I'll show that on Tuesday. But in general, and in this case too, we can solve the set of ODEs for the fixed points if we look for a dynamical systems analysis, and we see a steady state for each of the two phases. We can do the chemical master equation, and we see some change in the positions of these steady states. But then once we put it into the reaction diffusion master equation, you can see a continuous shift in the fixed points. So the fixed point positions depend on the diffusion coefficients of each of the molecules. So in the limit where you set those to infinity, you recover back the ODEs. But as you decrease them, in general, the position of the fixed points shifts too. So that does change. Yeah. And first passage time changes back and forth between the states. All depend on the diffusion coefficients. All right. Thanks.